Alright, everybody. Hello and welcome to Lecture 3 in Biochem 441. Today we're going to be covering DNA and RNA and their structures. This is the first lecture by Professor Gu, who has a interesting way of lecturing. I personally don't find it to be my favorite, but yeah, we'll go ahead and get into this, and hopefully I can shed some light on some of the stuff that was talked about, and we can, um, clear some of this stuff up. So, starting out, I'm just going to ignore all of this. There's all sorts of things about, biochemistry research and all that stuff, and how do you learn better and all that. But we're going to get into the content. That's what I really care about. So, the one thing I do like about Gu's lectures is the learning goals. And you can use these to kind of scaffold your learning and figure out what you need to work on. So we're going to start with explaining what genetic information is. So what is genetic information? This is the first thing we're going to cover right now. So to talk about that, we first have to talk about the central dogma and the genetic flow, or the information of that is genetics, and how that kind of flows from place to place, and from DNA into proteins that actually do things. So DNA, right, is, DNA has our DNTPs in it, our A, T, C's, and G's. And, when we're doing cell division, what we have to do is we have to replicate that. So when we're doing DNA replication, that's when we're creating new cells, and we need them to have the same DNA as each other. When we're going from the DNA to the RNA, that is transcription. And transcription, is when we start making RNA. And RNA is made up of A, U, C, and G. There's no T in RNA. The T's in replaced with U. So no thymines, just uracils. You can also go in the reverse direction. So reverse transcription is going from RNA to DNA. There's enzymes called reverse transcriptases, which you guys will learn about. They catalyze the reverse reactions. And I'm pretty sure this circular arrow here is meant to represent like RNA processing. It's a bit weird. I don't know why they've done that, but there's RNA processing as well. You have to add, like, in eukaryotes, for example, there's the 5' cap, the 3' tail, the poly A tail, and stuff like that. And after that, you go on to translation. And translation is going from RNA to protein. So you're going to take the different nucleotides, split them into codons, and those codons will be read off in threes, and every 3 bases is turned into an amino acid to make a protein that has a cellular function, like an enzyme of some kind, like hexokinase, right, from glycolysis or something like that. So for the key features of genetic information, the main thing I want to get across here is this idea of 3 bases. I don't know why he's made them circles, coding for 1 amino acid. That's kind of what this is showing. So, building blocks, structures before and after polymerization, and so this is like, we have our our RNA, for example, or whatever, and we're producing something from that. So in this case, maybe we're going from an RNA to a protein. Maybe that's what we're doing. And every 3 bases is 1 amino acid, and then we make our protein that way. He mentions these key words here. I don't know if he ever talks about them really that much. I would try and keep them in mind. Just remember that specificity, we're talking about like fidelity. How accurate is our machinery at making exact copies of DNA or at transcribing DNA, stuff like that. Diversification of sequences. How do we diversify? How do we introduce variation, genetic variation? That's going to be through like mistakes, right? You're going to want some mistakes to introduce genetic variation, maybe stuff like that. And then regulation is how do you regulate, right? So, yeah. And then this is kind of a rehash back to the previous image. Going from DNA to RNA, we use RNA polymerase, but then in the reverse direction, we use reverse transcriptase. And then from RNA to protein, you use the ribosome. And then if you're doing DNA replication, which you guys will also cover, you do DNA polymerase. So that's what genetic information is, right? Genetic information is just a TLDR. A way is just a way that things are organized, and it's stored in these nucleotides in these DNA molecules, and it's used to make proteins. And then that process is the central dogma, which goes from DNA to RNA to proteins. And if you want to go from RNA to DNA, you can. If you want to replicate your DNA, you can. And if you need to process your RNA, you can do that as well. So that's that learning goal done. Now we're going to go on to drawing DNA and RNA building blocks and like how those work. And I think I talked about this a little bit in the last lecture. We have a nucleotide. These things make DNA and RNA. The important thing to note is, I think you guys know this by now, but we've got our nucleosides, nucleoside monophosphates or nucleotide monophosphates. So we've got the monophosphate is just one phosphate, di is 2, and tri is 3. Makes sense. We got our pentose sugar. We've got some sort of base here. It's got to be A, T, U, C, G, something like that. Remember, U is only in RNA and T is in DNA. We have our glycosidic bond. If it is in DNA, it's going to be deoxy. It's going to have an H. If it's just a normal NTP, it's going to have an OH at this 2'. This 2' OH is very important for DNA structure and RNA structure and the different forms they adopt in water. So that's going to be very important to keep in mind later on. My throat's already starting to choke up. Okay. Now, we have our DNA structure. This is what DNA looks like when it is connected together. So, we have our pentoses, right? So our pentoses are these green circles here on this line. Our phosphates are the phosphate backbone, so these oranges, and the bases are the bases here. You should be able to tell me what all of these are, right? Without even looking. So I know it says a thing here, but you should be able to tell. Like, I know this is adenine, I know it's thymine, I know it's guanine, I know that's cytosine. You should be able to tell. And what you should know is, that's very important, is that these bases are actually very hydrophobic because they've got massive aromatic rings, while the outside is actually very hydrophilic, which makes sense because the phosphates are negatively charged. There's a bunch of negative charges on them. You can see them here. 1 minus here, 1 minus here, 1 minus here. A bunch of negative charges. So it's very negative, very hydrophilic. So the surface is going to be this area, is going to be hydrophilic, and the internal area is going to be, the axis is going to be, I guess the axis, what they call it? Yeah, the axis of rotation is what they mean by that, is going to be hydrophobic because of the rings that are there, these benzene rings, sort of, so to speak, or these aromatic rings. So, moving on, you can also see, I want to point this out, the 5' to 3' direction. Notice how this is the 5, this is carbon 5, this is carbon 3. It's kind of covered right here, but this is carbon 3 right here. Carbon 3. That's 3, that's 5. That's where we get the 5' to 3' direction from when it comes to our DNA. It just depends on it's just the name of the carbons of the pentose. Here we've got our nucleotides, nitrogenous bases. Already talked about this a little bit in the last lecture with Holt. Kind of sped through it a little bit. But yeah, you've got adenine, you've got guanine. Try and remember these structures. You need to remember these structures. You can put it on your note card if you want, but I personally think you should just memorize them. You've got cytosine, you've got uracil, you've got thymine. The main thing to keep in mind is at physiological pH, these things are usually uncharged and they're hydrophobic. At physiological pH. When you start altering the pH and, making it like pH 2 or pH 20 or something, then all that stuff goes out the window and you have to think about some other interactions that could be occurring. But at physiological pH, which is around 7.3 if I'm not mistaken or something like that, they're going to be uncharged. And because of that, hydrophobic interactions will dominate, and that's because of these massive aromatic rings that you can see. So this is something that you guys should have done in OChem. This idea here with the major tautomers, but tautomerization just refers to how some of these things can flip, basically. And an example of that is going to be like these here. So like there's lone pairs on. Let's see here. Like this here. So for example, you could maybe take an H off of yours, so this could come down here, and then you could make like a thing where you have like an OH on the bottom, and then you have an N lone pair here. So you can see how like the bonds gets flipped and stuff like that. That's what they mean by tautomers, and I'll show you that in a second. There's the amino and keto forms. And you also have to be able to identify hydrogen bond donors and acceptors on the not amino acids, the nucleotides. There's also Chargaff's rule. Chargaff's rule basically says that the number of A's should be equal to the number of T's, and the number of C's should be equal to the number of G's when you're looking at most species. Which makes sense, right? Every C should be paired with a G, and every A should be paired with a T. So that makes perfect sense. And also, you have to be able to identify the bases that are paired, so that should be paired. So C and G should pair, and A and T should pair. A and U can also pair. And the number of hydrogen bonds between them. So C and G have 3 hydrogen bonds, and A and T have 2 hydrogen bonds. So this is 3 H bonds, 3 HBs, HB, and 2 HB. 2 HB. Oh my goodness, bro. But yeah. And then Rosalind Franklin, cool. She discovered the helix, stuff like that. Great. So this is the idea of tautomerization. So what you can see here is it makes up 0.01% of a pool. So that's very little. So if you had, I'm trying to think of like how you would do that even. I think like if you had, I think it's 1,000 if I'm not mistaken here. Let's do a bit of quick maths here. If you had, I think it's 0.01 times 1,000, or no, 100. Oh no, sorry, wait, 0.01% is actually lower than that. 0.01 divided by 100 is what that is. I think it has to be like times 1,000 or 10,000. Yeah, no. If you had, basically, if you had 10,000 nucleotides, one of those is going to be one of these tautomers, essentially, and so that's kind of important to keep in mind when you're looking at these different DNA structures. How often does it show up? Not very, but it could. So something to remember. And then the way these tautomers essentially work is all you're doing is you're basically plucking off H. The electrons go down. This comes and grabs an H, and you end up with an extra thing like that. And you can see how that works there. For cytosine, it's going to be the same thing, exactly the same thing. We're going to grab this H, going to come down here. This grabs another thing up here. For this, it's kind of the opposite, actually. You're going to grab this H. This is going to come over here, and then you're going to grab an H from the solution. It should be H plus, by the way. I'm just too lazy to draw the H pluses, but... And then same thing here. You're going to grab this. This is going to come back. This is going to go up. This is going to go up and grab an H. But you get the idea. Basically, it's just a bunch of OChem reactions. But yeah, as it says here, actually, there's 3 billion base pairs. 300,000 could adopt the tautomeric form. Or 1 in every 10,000 bases. Now, here is just some information on Chargaff's rule. Like I told you, with Chargaff's rule, the A's and T's have to be equal, and the G's and C's have to be equal. That does not mean that the A's and T's combined have to be equal to the G's and C's. As you can see here, the ratio of A to G is not the same. So even though A to T is 1 to 1, G to C is 1 to 1, A to G is more than 1 to 1. So that means that there's just more A's and T's than there are some G's and C's. The other thing you should know is that if you get these kinds of numbers where the ratios of like A and T and G and G, A and T and G and C are not the same. Like if G and C are not the same number and A and T are also not the same number, it means you have single-stranded DNA. Because if you had double-stranded DNA, then they need to be equal to each other. The A's need to be equal to the T's, and the G's need to be equal to the C's. That is just a fact. It has to be true. If it's single-stranded, though, those rules go out the window. Those rules go out the window for Chargaff's rule. Does not apply anymore if it's single-stranded DNA. Totally could ask a question like that on your exam, so be aware of that, and be ready to answer a question on asking if it's a single-stranded or double-stranded DNA. So, yeah. Here we have X-ray diffraction patterns of DNA, and we've got the A form and the B form. And I will discuss these two forms in a minute, but you should also keep in mind A form, less humid, and B form is more humid. The B form is a lot better for base stacking, so the hydrophobic residues or hydrophobic rings on the nucleotides, those are much better. Those are much more stackable when you have the B form of DNA versus the A form of DNA, if I'm not mistaken. I won't bother with trying to explain, the early models and stuff like that. It doesn't really matter too much to me, and I don't think it matters too much to the class, so yeah. You really should not have to memorize these distances. Okay, these are not. You don't have to memorize these numbers. You can maybe put them on your note card if you want to be super safe, but I would be quite surprised if they tested you on that. But it's technically fair game for an exam. So, proceed with caution. So, for amino acids versus nucleobases or nucleotides, what you should know is that, if you remember from 440 when you memorized your amino acids, there are 20 amino acids, and there's a mix of a lot of things. There's positive ones, negative ones, there's polar, there's uncharged, there's polar uncharged, there's hydrophobics, there's all sorts of things. Very diverse. There's inter and intramolecular reactions. There's a whole bunch of stuff going on. With DNA, though, there's only 4. There's, technically 5, but for DNA specifically, it's only 4, not counting RNA. There's adenine, thymine, adenine, thymine, cytosine, and guanine. And those are all hydrophobic. All of them are hydrophobic. There's no ifs, ands, buts about that. So, and they're also very specific. With amino acids, I could put an A after a leucine, and then I could put an aspartate, and I could put a glutamate. Doesn't matter. With DNA, I have to pair it like this. I have to do A and then T. I have to do G and then C. And if I'm going to do a G and a C again, I got to do G and a C again. I can't mix and match really and do G and A. That doesn't really work. Unless you have a non-Watson-Crick base pairing. But DNA is very concerned. So, and then we have the phosphate backbone. The pKa of the first deprotonation of phosphate is about 1. So it's always deprotonated. It's always negatively charged. I am not going to go into this math stuff or the math here. I don't think this is very important. It's basically just calculating the number of bases per liter. But I don't think that's very important. So, These phosphates, though, they're negatively charged, and the way that you keep that balance is by using magnesium ions. So there's actually just magnesium ions floating around here. There's magnesium here, maybe another magnesium over here. You can imagine that there's a coordination complex forming between these magnesiums and the phosphates. And so the question becomes, why phosphorus? So just to make this very clear up here, actually, you can't just have negative charges floating around in space. That's why these magnesium 2 pluses need to be there to counteract those negative charges. And remember that this phosphate side here, this side is facing water, while this inner side here is facing into itself when in the double helix. So this side is very hydrophobic. This is very hydrophilic, and so this side is used to protect the inside from having to deal with water, essentially. And so the question is, like I said before, why phosphorus? And the answer to that is, well, first of all, you want to keep your DNA in a compact space in the nucleus, and the nucleus, is a membrane-bound organelle, so to speak. It's not, I guess, if you want to talk about it like that, it's an organelle, basically. It holds your DNA. And how do you stop things from going through membranes? Well, you make them charged, because membranes are hydrophobic and they do not like charged things. So phosphate at pH 7, very, very, very hydrophilic. It's ionized and it cannot cross the membrane. So DNA is not leaving anytime soon. It is stuck in the nucleus and it's never leaving. The other thing is that it is stable. And it's stable because phosphoester bonds have a half-life of 30 million years. Sorry, 30 million years. That is not breaking anytime soon. You would die about 8 million times over before, it's not even 8 million, it's way more than 8 million. So it's a massive number. You would die so many times before your DNA, half of your DNA, even degraded. Half of your DNA. It's crazy. So, very stable. And for synthesis cases, we know that, phosphates are really good leaving groups. ATP, stuff like that, they're okay with leaving. And, they release a lot of energy when they leave. So, very good for leaving groups, and they allow for easy synthesis of other molecules. They're also just very abundant in the environment in general. Like Earth has a lot of phosphates. Phosphorus is a very common element. Change phosphorus to arsenic. This is just a nice little study that they did. They tried to see if there were organisms that had, or if they could replace the phosphates with arsenic. Long story short, no, they can't. And the reason is because arsenic in water has a half-life of 0.06 seconds. And while, like I said before, phosphodiester bonds are 30 million years. So, yeah, you would be dead very quickly if you had arsenic instead of phosphates in your DNA backbones. So, no, you cannot use arsenic. That was kind of the whole point of this study. Okay. Now we move on to the chemical properties, the pentose rings. And what you should know here is for the rings, we've got the normal ribose and we've got the deoxyribose. And for the ribose, the only difference between these two is the 2'. There is a 2' OH on the normal ribose and a H here on 2' instead for deoxyribose. Now you might be wondering, okay, well, why the heck does that matter at all? And the answer to that has to do with the orientation of the molecule. So, you can imagine, for DNA, if we see here, which is deoxyribo, I would say that, oxygens in general, right? So you have to remember the 5 prime, right? This is where a phosphate is located. 3 prime is also where a phosphate is attached to. For the RNA, let's start with this RNA molecule, or the ribose molecule. No, it's not an RNA molecule. For the ribose, the 3 prime has this the phosphate, one of the phosphates. This is technically a P here, right down here. There's an RO group for the continuation of the chain, and there's another O up here continuing on. And there's also a O here from the fact that it's just a normal ribose, not a deoxyribose. When I have a lot of different electron carrying groups near each other, they're going to repel each other. And that repulsive interaction is what leads to these pucker conformations. And the different pucker conformations we have are C2 endo and C3 endo. And C2 endo, C2 prime endo, refers to the second carbon is being lifted upwards. That's what it means. C3 endo is just the 3 prime carbon is being lifted upwards. That's the only difference between the two. Now, the reason that we have these different forms is because of this interaction of this OH with these OR groups. And in the case of DNA and RNA, it's going to be this R is going to be a phosphate. So, and then there's also the 5 prime is also going to have a phosphate. So, let's look at this. If DNA, or if RNA, let's look at RNA first. If RNA adopts a C2 prime endo conformation, what's going to happen is that, what's going to happen I'm trying to they think they showed it here? Yeah, they showed it here. Yeah, so what's going to happen is, see how close this OH is to this OR? This is assuming that it's a normal ribose. These two things are going to repel each other. They're not going to be very happy with each other, and they're going to be very angry. And so because of that, the molecule would lose stability if RNA or riboses adopted this C2 prime endo conformation. So instead what you see is they actually end up adopting this C3 prime endo conformation. And that makes the distance between the OH and the OR significantly larger. It also makes sure that the distance between the 5 prime phosphate is also large enough where it's not causing too many problems. So the reason that the riboses generally adopt the C3 prime endo conformation is because of the fact that we want to make sure that this OH is not interacting with this OR group. And so if RNA and riboses in the RNA were to adopt this conformation instead, the issue would be that this OH group and this OR group are too close to each other, and they would repel, and that would cause your DNA to kind of self-hydrolyze and destroy itself. It wouldn't be very stable, that's for sure. But with the C3 endo, like I said before, because these things are now so far apart, that repulsive force is now less, and it's much easier to store your DNA in this form than it is to store it in this form, to have your RNA in this form than it is in this form. For DNA, on the other hand, though, that OH is not there anymore. This is not there. It's just a normal H. And so now all we're doing is we're trying to minimize the charge interactions between the 5 prime phosphate and the 3 prime phosphate, right? This 3 prime group. And the best way to do that, if you just have an H here, I'm going to try and erase this O here. There's no O here. The best way to do that is if you just put them on opposite sides. And that's why the C2 prime endo is the best for DNA, because, this OR is completely opposite to this phosphate and 5 prime. We couldn't do this before with RNA and RNA riboses, just normal riboses, because the phosphate here while it is also being repelled by the 3' phosphate, we also have to be concerned with the OH on 2'. And so that is why it's better for RNA to, lift this up a little bit to use the C3 endo instead. But for DNA, like I said, that 2' OH is gone. So it's totally fine. You don't have to do C3 endo here because you can just do C2 endo, move the 3' down, and you can have a much larger space between the 5' phosphate and the 3' phosphate. I hope that makes sense. This is a little bit weird. If you have questions, you can always leave them in the comments, or you can ask me at the IC. But hopefully that makes sense. The main thing to keep in mind here is that these different pucker forms come as a result of charge repulsion. So they come because, or they arise due to the fact that we need to make sure that our molecules and structures are stable, and electrons and like charges repel each other. And if that's the case, we have to make sure that the distance between our charged groups or our electron-rich groups is large enough to where it doesn't cause too many problems. And so in the case of, RNA and the normal riboses, we have 3 groups that contain this kind of like electron richness. And in DNA, we only have 2 of those groups really that we have to care about, which are this group and this group. And so here we have, like I said before, this just reinforces what I was talking about. See how close it's in the A form. See how close these phosphates are? These are very repulsive to each other. They are not happy. But in the B form, look at how far they are apart. They're much happier here. But then if I do end up adding an OH right here, that's electron rich, this phosphate is going to move there to that C3 position. See? I hope that kind of makes sense. But this is exactly what I was talking about. Role of 2' OH in favoring the A form structure. Steric clash with the backbone atoms. So, steric clash with backbone atoms. So you can see here, an example of this, right? This phosphate, like this gray thing here is the phosphate with these O's, is clashing with this 2' OH on RNA. And so what ends up happening is that 2' OH actually causes a self-cleavage reaction where this OH actually attacks this here and then breaks this off, and you end up with an RNA that catalyzes the hydrolysis of itself. The other thing to keep in mind is syn and anti. And when we talk about syn and anti, we're referring to syn and anti with respect to the phosphates. So syn is going to be on the same side as the phosphates, and anti is going to be on the opposite side of the phosphates. And the anti and syn are referring to rotation around this glycosidic bond specifically. So that's the only thing we actually really rotate. So syn, same side as phosphates, anti, opposite side of phosphates. And the rotation occurs around the glycosidic bond of the base. What you should know is that, generally speaking, a purine, so these larger ones, actually prefer the syn conformation. That's because in the syn conformation, they don't really have to worry about any kind of steric clashes. They're so large, and these groups are so far, like these groups that are like sticking out, like this nitrogen and this oxygen, they're so far removed from like the rest of the from like the phosphates and stuff that they don't really interact that much, and it's totally fine to have them in that way. On the other hand, though, for pyrimidines, you have to have them in the anti form, because if you put them in the syn form, what ends up happening is this C2 this O group here actually ends up clashing with, charge interactions with the phosphates and the ribose O. So For pyrimidines, it has to be in the anti form and not the syn form. And then for purines, it has to be in the syn form. So for pyrimidines, has to be in the anti form, and for purines, it has to be syn. The other thing to like, not other thing, but I guess that's like the main reason that we have to worry about syn and anti. For purines, it doesn't really matter for them necessarily. I think they can actually technically be both syn and anti, if I'm not mistaken. But yeah, generally speaking, if I'm not mistaken, yeah, they can generally adopt both. Generally speaking, it doesn't matter if this is facing this way or this way, if it's anti or syn. The interaction is essentially the same. So, yeah, it just depends on the type of DNA, though, if I'm not mistaken. Like, if it depends on the type of DNA, whether it's syn or anti for purines. But for pyrimidines, it's got to be the anti form, because of the fact that all the pyrimidines all the pyrimidines have this O group here, uracilthymine, and even cytosine. Cytosine doesn't have that O group there, but it's got an NH2 group. And because of how small that ring is in comparison to the purine ring, it just clashes a lot easier. But other than that, I hope these videos, or this video, not these videos, I hope this video was helpful. There's a lot of content to cover, and I know that, the explanations sometimes aren't the best, both in lecture and from me necessarily, but I hope this is at least somewhat helpful, and you can at least get an idea of what's going on. And, yeah. That is not what I wanted to pick up. Whoopsie. But yeah, thanks for watching, and hope you all have a good day. See ya.